All right. Hello, everyone. Welcome to the final lecture in our Science in the News Spring Series. We're glad you're here today. We are the co-directors of Harvard Science in the News organization. My name is Dana. And Science in the News, if you haven't been to one of our events before, or if you're not aware, we are a graduate student organization that has two goals. The first is to bridge the gap in science communication between scientists and non-scientists, and the second is to train graduate students like ourselves to become the next generation of science communicators. So we're glad that you can be here. We have some flyers, which Katie I think will tell you some more about, but over next to the flyers we also have a little questionnaire, feedback questionnaire. And if you could grab those either now or sometime before you leave today and just fill it out, your feedback is really welcome, both for how we run the event and also some feedback for our speaker if you'd like. We're always trying to get better at what we do, so we really appreciate it if you take a couple minutes and just write down any thoughts you might have. Great.
Yeah, so before I tell you about what's going to happen tonight, I'll just plug a couple events really quickly. So this is our last spring lecture. Hopefully you have stated your spring lecture needs.
If not, it's all up on our website, so you can rewatch them anytime.
That's true, but if you are dying for more Science in the News and you can't wait till our fall lecture series, we do have DayCon coming up, which is our free day-long science conference happening on Saturday, June 10th. It'll be in Maxwell Dorfin, which is right across the way. It'll be a full day featuring planet Earth as our theme, so we'll be talking about everything from honeybee migration to climate change policy to GMOs, water cycles, anything you can think of. And this day will be complete with breakout sessions where you get to actually hash out your own feelings on climate change. So come ready to discuss things and ask lots of questions. I do want to emphasize that the event is free, but you do have to register. So if you do want to do that, pick up one of these flyers or just go to sitinboston.com/daycon. And before we start off with Rebecca's talk, I just want to give you a quick overview of how things will be run tonight. So Dana and I will be here to moderate, but the show is entirely Rebecca's. If you do have questions, please hold them till our designated question breaks, which there will be, I think, 4 or 5 of throughout the entire lecture. We'll also have an intermission after part 2, where you'll get the chance to stretch your legs and grab more of our snacks. And you can also grab one of our surveys that Dana mentioned earlier and fill that out. It's a great use of 10 minutes. All right, I won't take up any more of Rebecca's time, but please welcome Rebecca Mann. She will be speaking on antibiotic resistance tonight.
Okay, so as we mentioned, my name is Rebecca, and I'm a graduate student at the Harvard Chan School of Public Health, where I study the disease malaria, which is a really important global infectious disease. But today I'll be talking to you about antibiotic resistance. So first we'll introduce the topic of antibiotic resistance by going over what exactly it is and how antibiotic resistance develops. And we'll discuss how this is really like a game of chance, where some bacteria just happen to get lucky. In part 2, we'll discuss some of the specific ways that bacteria can win this game of chance and avoid getting killed by antibiotics. But we'll also discuss the fact that the house rules mean that sometimes resistance actually comes at a cost to the bacteria. And finally, in part 3, we'll talk about some of the strategies that we can potentially use to help deal with the growing problem of antibiotic resistance. So starting off with the introduction. I'll first go over the public health implications of antibiotic resistance, and then delve more into the details of what antibiotic resistance actually means. So we all get sick, and sometimes when we have an infection, we're given antibiotics to help treat it. I'm sure everyone in this room has had this experience at some point. And specifically, antibiotics are medications which are very effective at treating diseases that are caused by bacteria. But unfortunately, sometimes these bacterial infections are not responsive to the medications that we typically use to treat them. So one of the most notorious examples of this, which you might have heard of, is methicillin-resistant Staphylococcus aureus, or MRSA. So we can't treat MRSA with methicillin, and a lot of MRSA is also resistant to other antibiotics that we may be able to use against it. So, MRSA. So because antibiotic-resistant infections such as MRSA are much more difficult to treat, they typically lead to a higher likelihood of complications and even death for patients that are acquire these infections. Altogether, the Center for Disease Control and Prevention estimates that antibiotic-resistant infections, such as MRSA, are responsible for over 2 million illnesses in the United States and lead to about 23,000 deaths each year. The global numbers for antibiotic resistance are not known, but it's estimated that many countries have even higher burdens of antibiotic-resistant infections than the United States. We also know that antibiotic resistance is a growing problem. So these two graphs both show the trend of increasing antibiotic resistance over time. The graph on the left shows the increase in bacteria that are resistant to vancomycin that emerged in American intensive care units, whereas the graph on the right shows the increase in hospitalizations that are due to that MRSA infection that we talked about. So imagine that these trends continue and that over time we are no longer able to use the drugs that we have to treat or manage infections. In such a scenario, it's estimated that resistant infections could cause up to 10 million deaths per year. This is more than all of the deaths caused by all types of cancers combined. So now that I hopefully have your attention, let's delve a little more into what antibiotic resistance actually is and how it develops. Firstly, I've already mentioned that antibiotics are medications that we use to treat bacterial disease, but I want to quickly make a note that not all diseases are caused by bacteria. So other important microbes that can cause disease include viruses and parasites. I won't really go into the differences between these different types of microbes, except to note that they cause different types of diseases. And I've listed some common examples below. So again, only diseases that are caused by bacteria, like strep throat or staph infections, are diseases that we can treat with antibiotics. I mentioned to you at the beginning that I study malaria, and you'll see that that's actually a disease that's caused by a parasite. And the other thing I want to mention is that a lot of common diseases that we get, that most of us have experience with, things like the cold and the flu, are actually caused by viruses. And these diseases should not be treated by antibiotics. No, it's super miserable if you have just a really bad cold. Please don't go to your doctor and ask for antibiotics to treat your cold or your flu, because they won't actually help you. And in fact, as we'll talk about a little bit, the unnecessary use of antibiotics can actually contribute to the spread of antibiotic resistance. So now that we've covered antibiotics and what infections we can use to treat them, let's define the concept of antibiotic resistance. So I first want to test your knowledge and pose the question, is antibiotic resistance a property of the bacteria or the human body? How many people think it's a property of the bacteria? Raise your hands. Okay, you guys all get A pluses. That's correct. Antibiotic resistance is a property of the bacteria, but it's actually a really common misconception that it's a result of the human body becoming immune. So this is an incorrect misconception that a lot of people actually have. And so it's important to know that antibiotic resistance is a property of the bacteria, and this actually has some really important implications, because this means that actually anybody can become infected with antibiotic-resistant bacteria. This is not just a problem that impacts people who have taken a lot of antibiotics in the past. This also means that antibiotic-resistant infections can spread from person to person. And because of this, we all have a responsibility to prevent the misuse or the overuse of antibiotics, and as I mentioned, to help prevent the spread of resistance. So antibiotic resistance, importantly, is a property of the bacteria, and it's the ability of the bacteria to survive in the presence of drugs that normally kill it. So how do bacteria develop this ability? In a given population of bacteria, there's going to be some random diversity. So just like you're different from the person you're sitting next to, no two bacteria are created exactly equally. And this is due to genetic variation or differences in our DNA. I'll go into that in a bit more detail in a little bit. But just normally, these differences may not have much of an impact. The green bacteria might not do better than the blue bacteria. However, if this population is exposed to antibiotic, by sheer luck of the draw, some bacteria might happen to be able to survive and not get killed with the antibiotic. So in this example, the red and the blue guys can survive while all the other bacteria are killed off by the antibiotic. And in this game, it's winner take all because the rest of the bacteria have died off, so the two bacteria that are resistant are able to quickly replicate and take over the entire population. So now you have a whole population of resistant bacteria. So this is a big picture overview, but where does this genetic diversity that I mentioned come from in the first place? This may be reviewed for some of you, especially if you attended the lecture a couple of weeks ago, but one important source of genetic variation is through random mutations. So just as a brief review, DNA is a molecule that's found in all cells, both our cells and bacterial cells, and it acts as like an instruction manual for the organism. It's made up of different bases, which are like the letters that make up these instructions. RNA is a similar molecule that gets copied from DNA, and then the RNA gets made into proteins, which can be thought of as the building blocks of the cell. Proteins have various functions inside the cell that are essential for the cell's survival. If there's a mutation in DNA, as indicated by this base in red, it goes down the line and also causes a change in the protein. So that's one important source of genetic differences within bacteria, just random changes in DNA. And I'll also note that while both our cells and bacterial cells contain DNA, one important difference is that the bacteria typically have one circular chromosome or unit of DNA in their cells, whereas you may know that humans have 23 chromosomes, which are linear. So you can probably get the sense that the DNA in bacterial cells, which is also known as their genome, is smaller and less complex than the human genome. However, unlike us, bacteria can do this really neat trick where they actually swap bits of DNA between each other. This is known as horizontal gene transfer. And so, for example, some bacteria are actually able to take up DNA from the outside environment and integrate it into their own genomes. Horizontal gene transfer can also occur during this direct cell-cell contact. And in this case, the bacteria are actually exchanging these smaller pieces of circular DNA, known as plasmids. So these plasmids are separate and independent from the genome. They replicate independently from the rest of the bacteria's DNA. And the bacteria can actually transfer one copy of this plasmid to another bacteria. So now both bacteria each have a copy of this plasmid. So to quickly review, the sources of genetic variation that we've talked about are random mutations in the DNA that lead to changes ultimately in the proteins, the uptake of DNA from the environment, and the transfer of plasmids between bacteria. So now that we've learned about how genetic diversity occurs in bacteria, I want to return to this bigger picture. You may have noticed that this is actually a perfect example of natural selection or survival of the fittest, where we have genetic differences within a population leading to some bacteria being able to survive and reproduce better than others. And in this case, we refer to the use of antibiotics as a selection pressure, because it's actually selecting for or screening out specifically the bacteria that are resistant, in this case, the red and the pink bacteria. And this is exactly why, while it's the genetic variation in the population that allows some bacteria to be resistant, it's actually the use of antibiotics that facilitates the emergence and the spread of antibiotic-resistant bacteria. And so this is why it's really important to not use antibiotics when they're not necessary, because the more we use antibiotics, the more we're potentially screening for these bacteria that are resistant and able to survive the antibiotics. Right, so again, to highlight this point, the use of antibiotics is ultimately what leads to this emergence of resistance. So I told you that antibiotic resistance is a great example of evolution in action, but when we think of evolution, we might actually think of something like this, very gradual change that happens over millions of years. But in contrast, antibiotic resistance happens very quickly, sometimes in a matter of days. So to highlight this, here's a timeline of some of the antibiotics that are used clinically. So for example, the antibiotic tetracycline, which I'll talk about later in the talk, was first introduced in 1950. And resistant bacteria were first detected 9 years later. Methicillin was introduced in 1960, and that methicillin-resistant staphylococcus, or MRSA, that causes such a problem, was detected just 2 years later. Not to be outdone, when the antibiotic levofloxacin, which the brand name is Levaquin, was introduced in 1996, bacteria resistant to that antibiotic actually emerged within that same year. Ceftaroline, or Teflaro, is a new antibiotic that was introduced in 2010, and it was actually specifically designed to be able to kill bacteria that were resistant to some of the older antibiotics. However, ceftaroline-resistant staphylococcus bacteria have already emerged just 1 year later. Penicillin is a really interesting example because penicillin was first used clinically in 1943, but bacteria resistant to penicillin were actually first detected 3 years earlier in 1940. I'll talk a little bit more about why that is in the second part of the talk. So how is it that this process of evolution, which takes millions of years in organisms like humans, happens so quickly in bacteria? Well, let's consider that in humans, generation occurs about every 25 years, whereas bacteria can replicate themselves somewhere between every 20 minutes to 24 hours. And this very fast replication of bacteria means that they have population sizes that are much, much larger than humans. So the entire population of humans on Earth is around 7 billion, but the number of bacteria in a single human body is estimated to be around 39 trillion. And we can think of evolution of resistance as sort of this lottery, this random lottery where we have these mutations and this taking up of DNA that's happening at random. And actually, most of the genetic changes aren't necessarily going to be beneficial to the bacteria. However, even though this is this game of chance that, just like most of us have not very much of a chance of winning the lottery, most bacteria aren't going to have that chance to become resistant. Bacteria always win this game, and that's exactly because they replicate so quickly and they have such large population sizes. So all it takes is that 1 in a million or 1 in a billion resistant bacteria to get lucky and to be able to survive. So now that we've learned all about the evolution of resistance, let's actually watch it in action. This is a video that shows bacteria becoming antibiotic resistant over a long time. You'll see the bacteria growing in what's called agar, which is a common material that we use in the lab to grow bacteria, except for what we usually use sort of smaller plates. This is a gigantic agar plate, and it's really cool. One term that they use in the video is the term mutant, and that just means a bacteria that has gained a mutation. This comes out of the labs of Roy Gashoni, who's at joint appointment at Harvard Medical School, and the Israel Technical Institute. All right, let's watch it. Sorry. Full screen.
I have to try and go over it.
Oh, shoot.
So what we ended up building was basically a petri dish, except that it's 2 feet by.
Yes. All right, there we go. Cool. Okay.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band is 1,000 times as much antibiotic. And then across the top of it, or some thin agar, that bacteria can move around it. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. Then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Was that cool? Was that scary?
Yes.
All right, how do I exit this? Right, so in that video, you saw an example of how these individual genetic changes in the bacteria allow them to eventually survive at higher and higher concentrations of the antibiotic. And hopefully this gives you a sense of how this process can occur really quickly, since it took just 11 days for the bacteria to reach the middle of that plate. So to summarize part 1, hopefully I've convinced you that antibiotic resistance is a significant and growing public health problem. We've discussed how antibiotic resistance is a property of the bacteria, which is a really important point. And then we've also talked about how the different ways that genetic variation can occur in bacteria, either through mutation or horizontal gene transfer, where they swap bits of DNA. We've discussed how antibiotic resistance emerges due to the natural selection of resistant bacteria, so natural selection because of antibiotic use. And we discussed how evolution is a game of luck, but a game that bacteria get to play millions and millions of times over. And with that, I'll take any questions for this part of the lecture. Yeah.
So you said you should avoid antibiotics if possible. And I understand that for taking antibiotics, but what about like antibacterial soap? Is it not good for use? Is that?
Maybe you
They
could
just banned triclosan.
repeat and or summarize the question.
Okay. Yeah, so the question was about the use of antibacterial soap. That's a great question. I think there's sort of different arguments, and I think like there's different types of antibacterial soap. So my understanding is like there are some that actually contain like chemicals that are like antibiotics, and there's some that just kill bacteria through just like using like ethanol or other more general chemicals. And so I think like, is that your sense?
So we actually have an article about this if you want to go to our website, written by Gabe Rangel, who is one of our spring series directors. But just to summarize that article for you, basically the FDA recently banned, I heard it over here, that they recently banned triclosan and a few other compounds that are antibacterial compounds in soap. So you'll see those disappearing off shelves in the next few months, basically because, not because they're harming us necessarily anyway, but because there wasn't strong enough evidence that they were doing additional good above and beyond just having soap, so detergents in our soap, and the rubbing motion Those two things, the soap and the rubbing motion, running your hands under water and doing this, are the main things that get rid of harmful bacteria and dirt and all that stuff on your hands. So keep doing that and you'll be fine. But that's kind of the consensus there. They are testing those compounds right now to see if there is added benefit, but you probably won't expect to see those back on shelves for quite some time.
Any other questions?
So now that we've gone over how the development of antibiotic resistance is like this game of chance, now we're going to talk about, again, some of the specific ways that bacteria can win this game and avoid getting killed by antibiotics. And these specific strategies that bacteria can use are known as resistance mechanisms. We'll also talk about how some of these resistance mechanisms can actually come at a cost to bacteria. So to talk about how bacteria avoid being killed by antibiotics, we have to know first a little bit about how antibiotics work. So we already discussed how proteins are molecules in the cell that have important functions. And so antibiotics typically bind to proteins in the bacteria cell and prevent them from doing whatever their function is. And often these antibiotics target a particular type of protein called an enzyme. And you can think of an enzyme kind of as a molecular machine which performs some particular activity in the cell, like making molecules. Now, if you think about how you want to kill a bacteria, you probably want to target some process in the cell that's really essential. So a lot of antibiotics actually target this process that we talked about, going from DNA to RNA to protein, because what could be more essential than that? It's a process that's fundamental for all of life. So for example, the antibiotic rifampin specifically targets the enzyme that makes RNA based on the DNA template. Similarly, the antibiotic tetracycline prevents the production of proteins from RNA. Another really important and common group of antibiotics are called beta-lactams. And beta-lactams target a protein that's important for building the cell wall of the bacteria. And so when the bacteria are treated with these beta-lactam antibiotics, their cell wall isn't as strong and they'll actually burst open, which you can imagine is also a pretty effective way to kill them. So these are some of the ways that antibiotics can kill bacteria, but you already know that bacteria have developed some ways around this. One of the most common ways that bacteria can become resistant to an antibiotic is by changing that protein that the antibiotic or the drug is actually targeting. So normally the antibiotic is specifically designed, as we talked about, to bind to this particular bacterial protein and stop the protein from doing its function, which ultimately kills the bacteria. However, as we discussed, random mutations occur all the time, and sometimes one of these mutations can actually happen in the protein and change the shape of the protein so that now the antibiotic can no longer bind and that bacteria is resistant. For example, the enzyme that I talked about that produces RNA from DNA is called RNA polymerase. And I told you that rifampin is an antibiotic which disrupts this process of going from DNA to RNA. So rifampin actually is specifically targeting this RNA polymerase. And that makes it a really good drug actually for the treatment of a lot of bacterial diseases. But unfortunately, a single mutation in the RNA polymerase can prevent rifampin from binding and thus lead to resistant bacteria. Another way that bacteria can avoid getting killed by antibiotics is to decrease the amount of antibiotic that is present in their cells. Bacteria can do this in two ways. First, it can just block the antibiotic from entering in the first place. So oftentimes, antibiotics aren't able to enter directly through the bacterial cell membrane, and they have to come through some sort of channel or pore in the cell. However, a mutation in this pore can prevent the antibiotic from entering through the pore. And in some cases, the bacteria might just get rid of the pore altogether, and now the antibiotic has no way in. The second way that bacteria can decrease the amount of antibiotic they're exposed to is by actually actively pumping the drug out of the cell. And this is one of the primary ways that bacteria can become resistant to tetracycline, which I mentioned is an antibiotic that targets the production of protein to RNA and blocks this process. Tetracycline pumps in bacteria specifically bind to this drug and are able to pump it out of the cell. And tetracycline pumps are often found on plasmids, or they're those small pieces of circular DNA that we talked about, which can transfer between bacteria. And so this mechanism of resistance of pumping the drug out of the cell can actually get spread from bacteria to bacteria and thus can rapidly work its way through a population. Also, while there are some pumps that are specific for particular drugs, such as tetracycline, there are also pumps that are multi-drug resistant, so that they can actually bind and pump out a variety of different antibiotics and protect the bacteria from being killed by multiple types of drugs. The last strategy that we'll talk about that bacteria can use is to actually degrade or inactivate the antibiotic. So I mentioned that enzymes are proteins which perform specific functions within the cell, and in this case, bacteria can have specialized enzymes whose job it is to break down certain types of antibiotics. So one important group of these enzymes are known as beta-lactamases. And beta-lactamases are enzymes in bacteria that specifically degrade beta-lactam antibiotics. So those are the antibiotics I mentioned that prevent the formation of the cell wall in bacteria. And some types of beta-lactam antibiotics you might have heard of include penicillin and cephalosporin. And you can probably notice that looking at these chemical structures is that they both have this square-shaped structure that's highlighted in red. And it's this square, it's called the beta-lactam ring, that the enzyme actually is able to recognize and sort of attack and break apart, thus inactivating the antibiotic. Now, you may remember that I also mentioned that resistance to penicillin actually emerged before penicillin was used clinically at all. And the reason for this is that not all antibiotics are actually made by humans. Other microbes, actually, like fungi and even some other bacteria, can also produce compounds that are antibiotics. And you may actually know that penicillin was first isolated from a fungus that Alexander Fleming found in his lab and noticed could kill off bacteria. So different microbes, bacteria, and fungi are producing these antibiotics to actually kill off each other. Microbes are engaging in this sort of chemical warfare, and in response to this, bacteria have developed these different enzymes to break down these chemicals that bacteria and microbes are throwing at each other in order to protect themselves. And the evolution of these enzymes happened long before humans came into the picture. So in an attempt to thwart this, we as humans have created newer drugs, third generation drugs they're called, which were designed specifically so that they are more complex and they can't be broken down by beta-lactamases. However, bacteria can actually gain additional mutations in the beta-lactamase enzyme, which provides them with protection against even these newer antibiotics. So to briefly review, the different ways that bacteria can avoid being killed by antibiotics include changing the protein that the drug is targeting so the drug can't bind, preventing the drug from entering by changing or deleting a pore, actively pumping the drug out of the cell, or using an enzyme like beta-lactamase to degrade or inactivate the drug. One very central concept, which has been studied extensively, is that the evolution of resistance often comes at a cost to bacteria. So in exchange for gaining some of these resistance mechanisms, bacteria often have to pay up. And if you think about it, this actually makes sense, because I just told you that becoming resistant often means making changes to important components of the cell. So, for example, if a bacteria gets a mutation in a target protein, this may make them resistant by no longer allowing the drug to bind, but it could also make those proteins function less efficiently in doing whatever task they're originally supposed to do. So, for example, we talked about rifampin-resistant bacteria with mutations in the RNA polymerase. And multiple studies have looked at these resistant bacteria, and they've actually these studies are actually able to measure RNA production in the lab. So this is a study that comes from Mycobacterium, so it's the bacteria that causes tuberculosis. And what this data shows is that the resistant bacteria that have those mutations in RNA polymerase produce RNA less well or less efficiently as the sensitive bacteria. So these resistant mutations are having an effect on the bacteria. You can also imagine that loss of a pore, while it may prevent antibiotics from getting in, might also make it more difficult for the bacteria to get the nutrients it needs from its outside environment. And even sort of new genes that can be acquired, such as the beta-lactamases or the pumps that we talked about, can also come at a cost, because they just require more energy and resources to make and maintain these brand new proteins. Thus, while resistant bacteria, as we talked about, are this huge advantage to the rest of the bacteria around them, where there is antibiotic present, in the absence of that antibiotic pressure, resistant bacteria can actually be at a disadvantage. So, for example, that same study looking at the rifampin-resistant bacteria also showed that the resistant bacteria don't grow as well as the sensitive bacteria. So these are two auger plates, kind of similar to the video that we watched earlier. And you can see that there's these circles, and these circles are actually growths of bacteria. So there are multiple bacterial cells in each of these circles that have grown up. And you can see that, we call these colonies. And you can see that the colonies of the rifampin-resistant bacteria that have those mutations are much smaller than the sensitive bacteria, indicating that they're growing less well. So resistant bacteria can have these disadvantages, and because of this, resistance often leads to what we call a fitness cost, which is a decreased ability to survive and replicate. So to think about the impact of a fitness cost and what this actually means, let's imagine an experiment where we take two flasks, one with resistant bacteria, one with sensitive bacteria, and we mix them together. Now, we already know that if we treat this mixed population with antibiotic, the resistant bacteria will quickly take over the flask, because they're the only ones that can survive. But what would happen if we just let the bacteria in the flask grow on its own without antibiotic? If there is a fitness cost, then what we'd expect is that over time, the sensitive bacteria would actually start slowly taking over the flask. This type of experiment is called a competition assay, and it's often performed as a way to actually measure fitness in the laboratory. And in fact, that same study of the rifampin-resistant bacteria that we've been talking about actually showed that the resistant bacteria do have this fitness cost, and so rifampin-sensitive bacteria are able to outcompete in an experiment set up similar to this. So to summarize part 2, there are a few main ways that bacteria are able to decrease their susceptibility to antibiotics. And this includes changing the target of the antibiotic, degrading the antibiotic, or decreasing the amount of antibiotic in the cell. We've also talked about how the genetic changes that lead to resistance often comes at a cost that can eventually impact the fitness of the bacteria. that, I'll take questions for part 2.
Are we able to employ any bacteria to fight the resistant bacteria?
Well, we've definitely, sorry.
Repeat the question.
Oh, right. So the question is, can we employ other bacteria to defeat resistant bacteria? It's a really interesting question. I only know that, we do sometimes some of the antibiotics that we use are originally discovered from bacteria or fungi, and we figure out that they're able to kill, and we figure out how to synthesize those compounds ourselves. As far as sending in bacteria to outcompete other bacteria or something like that, not that I know of.
So I can answer this one too. So one way that I think is amazingly cool is called predatory bacteria. There are 2 genuses of bacteria. One of them is called Vibrio, and as you can imagine, these are actually vampiric bacteria that can actually latch onto other bacteria and suck out their insides and kill them. And they actually have found that some of these strains of bacteria can be used to kill antibiotic-resistant bacteria. The problem is we have to make sure they're not also killing the good bacteria inside of us. So they're developing ways to use these bacteria as tools to kind of fight fire with fire.
I think the other kind of related therapy that's interesting that I think has been used clinically is phage therapy.
Yes.
So phages are viruses that can attack bacteria. There's always sort of an issue with these strategies, of course, is people are a little bit wary of taking a pill that contains bacteria or viruses. That sounds a little scary, so that's sort of, and then there's a lot of safety concerns. So but it's
Just
really
call it
interesting.
probiotics.
Yeah.
You mentioned something about, when you were talking about the different mechanisms that we pump it out strategy tends to show up on these plasmids or whatever, the little shareable DNA chunks. And I was wondering, is there a natural reason why that kind of resistance would make sense to be on the plasmids, whereas some other thing, is it because that's a sort of modular, a pump is something you could add to any bacteria maybe, but a new version of your RNA polymerase protein, you can't really get that on a plasmid? What's the connection?
No, it's a good question. I don't think it's entirely known, necessarily what the first origin of these pumps are. My sense is that one reason is that it can be on a plasmid is that it's not actually essential for the bacteria. So you wouldn't want an essential protein to be on a plasmid that you could accidentally lose. That would be bad. And sometimes with plasmids, if the bacteria doesn't need them anymore, it can either sort of stop making the proteins in the plasmid independently of its genome, or it could potentially lose the plasmid over time. So there's, in these cases of proteins that the bacteria only needs in certain situations, my sense is that that's why it's sometimes a bit more advantageous for them to be on the plasmid. It's a really good question. That's just a general sense. I haven't looked into it really specifically, but I think that would be sort of the general reasons why. And again, I think, as I said, the exact origins are not fully understood. Yeah.
When the something in the different bacteria are in the same class, how do they recognize the other type of bacteria?
Right. So in that experiment, it's not that the bacteria. So in this case, they're all the same type of bacteria. They're not actually attacking each other. They're just sort of growing normally, but you can't have an infinite number of bacteria in the class. There's sort of limitations of nutrients, and so it's just sort of a process of evolution where some will be growing more quickly and some will be growing as quickly, and so you'll just over time have the sensitive bacteria take over. And it's not that they're fighting against each other necessarily. They're just sort of growing along, and sensitive bacteria are just growing better. Yeah, good question for clarifying that. Okay. With that, we'll take intermission.
Yep. Feel free to grab some more snacks or a survey if you'd like. And we'll be back in 10 minutes.
Do you have that flyer for day on?
I do, yeah, there are a bunch over here.
This one is.
Thank you.
Yeah, so.
So bacteria.
Is most of the random changes through like gene multiplication or something like that? Or like what is the?
My sense is, and so I'm a pastologist, Katie's actually a bacteriologist, so she can confirm this, but my sense is that that can actually differ from bacteria to bacteria, right? Like whether mutations or gene complications, like might depend on the species.
So the most common way for mutations in genes to occur, so when bacteria replicate and produce more of themselves, they have to copy their entire chromosome over. So instead of using RNA polymerase, which is for like making actual products from the genes, they're using something called DNA polymerase, which is just making like a carbon copy of the instructions to the cell. And that's where mistakes happen, like while it's copying over. There are kind of troubleshooting mechanisms for it to like check what it's just written over, but sometimes it makes mistakes. And if those happen to create changes, then that's how, the competition.
But like, so for example, on the chromosome, you have maybe duplicate spots, and then one can change, but it won't necessarily, maybe it makes the new protein, maybe it doesn't necessarily screw the protein you need, but in that case, you still already have the other spot making the correct protein. Is there something?
Oh, okay, so there's only gonna be one chromosome per bacterial cell. So when it divides, each of them gets one chromosome. And so like the old one is intact. If there's a mistake on the new one, that cell has to live with that for the rest of its life. And all of its daughters,
Yeah.
same thing. Yeah. Did that answer your question?
Yes, but you're probably speaking like we have here, and I'm.
Okay, good. Please let me know what I can clarify.
Oh, I have lots of questions.
That's what we're here for.
Are you familiar with Sarah C. Smith?
So,
She's been working with antibiotic resistance for a while. She's a good communicator on Facebook and Twitter.
Check her out.
She's been interviewed on the Skeptic Skyler Universe and places like that. So I think she's still. I'm trying to think where she's at.
lots of bacteria involved.
Huge population.
Big colonies.
I didn't know at which point I asked this question. Is there any guidance that you give? Because now with outcome-based care rather than just number of procedures, and then also the push for patient satisfaction, especially in hospitals, our doctors, well, at least from the few I know, are saying that parents are demanding like antibiotics. And I remember when I was a kid, you didn't talk about this. You got a cold, whatever. You went away with a amoxicillin or a Z-Pack or something like that. And it was like, and I found that when I went to like the urgent care centers, that was just like, and you get this for free. This is, so how do you change that out of culture where you go, you get the antibiotics, the virus just runs its course, and you get better anyway. But now you think I need that, and then when you go and the doctor says no, you're dismissing right now. I'm the patient.
Right. Yeah, I think that's one of the biggest problems. I think educating both doctors and patients about this, because I think there are a lot of doctors, especially those who are used to giving the antibiotics out like candy. It's hard for them to change those habits. They see someone sneezing and they're like, go home. But
We're hoping that, through things like this and just trying to push more headlines are reasonable about antibiotics. Kind of the best we can
CDC
do.
has a lot of posts about this, and it's always hilarious in the comment section, because you'll get people screaming about them pushing vaccines or things like that, and you're like, you are complaining about them telling people not to buy a product, and they're still complaining to them.
Well, one thing I've actually read
And then you have
about,
the chronic Lyme people show up.
one thing I've read about actually in terms of like sort of antibiotics and changing behavior is the one thing that like kind of seems to work better is by emphasizing the potential, the bad effects of taking antibiotics, the negative side effects, because it's hard to really grasp this like big picture future implications. But if you say like you don't need this, and if you take it, it might give you some other like bad things, like bad effects, maybe you sick.
Well, let the flux and you can snap a tendon.
Right. And I mean, like, I don't know how often you've taken antibiotics in your life, but like a lot of times, if you take a really like powerful antibiotic that hits a lot of different bacteria, you have like diarrhea. Because it's killing all the bacteria in your intestines.
All this stuff too.
Right. It can't say like, hey, you're not supposed to be here. It just goes. And so I think that's a really good point. Like remind people that like this is killing a lot of stuff.
I think that there was also more recent that under the age of 1, you would want to avoid it at all costs because they're developing the microbiome through contact with breast milk and the areola actually, but then also just skin-to-skin contact, or even just the microbiome on your skin.
Yeah.
And all people think it was good.
Yeah. I mean that they actually know that the microbiome keeps developing through like age 16 and 10. So that's why we really don't want to be giving like, antibiotics. It's so different. Because I remember when I was a kid, I took antibiotics all the time. And my dad was a doctor. Like, no one knew any better.
Yeah.
But I'm really glad you brought up the point about the FDA.
The CDC.
Well, no, the earliest someone said something about trichosan. Is that?
I said trichosan, yeah.
Yeah. So,
They put that out and gave companies till 2018. They started talking about this in 2015.
So they didn't talk as much about that because there's not as much. If you do want to check out that article,
People were saying that it was an endocrine disruptor was the concern. We never really found that unless you used it a lot.
right? Like there was like sufficient concern about potential bad effects and not enough evidence to back up the potential good effects that they just decided to better say I'm sorry.
Because it was like 99% versus like 99.9%.
Yeah, exactly.
Potentially no events or something. Right. Awesome. Okay, I'm gonna switch for your side. DDT. Back. And let's do the entire room. Go for it. All right. Okay, everyone knows, science pretty well. They kind of just threw it out of proportion in short, which is it's still the environment, but like in terms of the potential damage to humans is actually very low from what I understand, especially when you use like inside the house where it's not going to reach the watershed. Is that something? Think about that if people have been more reasonable about how many millions of people in that time period. Is there anything involved with that?
Well, there's not really effective like insecticides now that they're using, I forget. But of course, the other issue with insecticides is that the mosquitoes can also get resistance to them. So that's like a really active area of research.
I heard that they're developing a vaccine.
Yeah, it's not very good.
Yeah.
It works, but it's partially works. It seems to my PI actually like since then involved with those efforts. Although it's not like directly my project, but like there's been evidence coming out that it's like more effective against some strains or types of parasites than others, and that its effectiveness is it might do something. It probably does something that's fairly commercial.
Now, with
Do we have to? Sorry.
We got a couple minutes left.
I will not. So, for example, I know a Is the malaria itself somehow some like a toxin mediated, like where it's just it's waste product just builds up?
Like how the
botulinum, releases the toxin, and that's what gets us, not bacteria proliferating.
This is actually malaria, it's because malaria proliferates in our red blood cells.
Okay.
And then it also can get into the brain, so it's not supposed to. Like if you get malaria in your brain, that's really bad. So it's actually
Is
like
it
the bacteria.
mutation? That's the problem.
It's replicating in blood cells.
So it's
Yeah,
stealing
so
oxygen?
It's destroying blood cells. It can like
Oh, it's destroying blood cells.
it can mess up your liver pretty badly.
Like full on apoptosis of red blood cells.
Yeah, when it replicates and then it bursts out of the blood
Yeah.
cells. So
Okay.
those red blood cells are destroyed. So people can become anemic.
So how would you target that? The vaccine, like you can't use a self-immune vaccine because it's just you're throwing up proteins to be alive anyway. Like what would you, how would you, that's what I would.
So
wonder how you
not
approach that.
so that's a great question. I guess with vaccines, there are several kinds of vaccines. You mentioned a couple. So some are where you take whatever you're trying to get rid of and you make like an version of it, a dead version of it, and you put that in, the immune system recognize that as a function of the actual harmful. Others will actually just like, so the way our immune system recognizes those things in the first place is they'll say, hey, you have something on the surface of your cell that looks nothing like what's on the surface of your cell, so we're gonna get rid of you. If you just take those proteins or those little molecules and you put them into your it'll still be a very foreign thing.
Yeah.
And that can also affect your immune system. So that's the idea behind a lot of vaccines. You don't need to be putting in like a full malaria parasite.
Okay.
I don't actually know exactly how the malaria vaccine
It's all good.
works.
It's okay.
Rebecca,
It
what
was
does
gonna
the malaria
happen.
vaccine recognize?
It's a specific protein that's on. So there's different forms of the parasite as opposed to the body. So the form of the parasite in the skin first injects. It's like swimming before it reaches its cells and has these proteins on the surface, and it's those proteins that But yeah, there's different sort of other candidates too, and actually the makes other proteins get expressed on the surface of the red blood cell that would normally be there. So there are still options for the protein-based vaccines.
Because like something with like, and this is bacteria obviously, but like something with the Lyme disease one, you have to have enough of the antibodies to actually get into the tick to kill the spider bugs before they even develop the tick into your body. So I don't know how that one operates either. And the first one, both the first one you're talking about, where the T cells and the macrophages and the T cells are working together. Like that's what I'm wondering, like, is there a part of the parasite itself? Because I know very little about parasites. It's hard to tell. Outside protein, does it get into the parasite?
Usually
And it makes it, like, not shut on. Exactly. Our immune system attacks.
So
Well, the parasite.
they have a lot of parasites because parasites are bigger than bacteria because they are so small. They'll usually recognize them from the outside of the parasite so that like
That's how you get
something destroyed will be marked and then destroyed. Or like I said, if it's
Okay,
inside the
sorry.
it looks different to be higher red blood cell.
Katie.
Lots of discussion going on.
So just fake just like
Exactly.
All right. So welcome back. So now that we've discussed this really enormous potential that bacteria have to evolve resistance, as well as the really great variety of ways that they of strategies that they're able to use, let's discuss whether will the odds be ever in our favor. And specifically, can we use our knowledge of how resistance evolves to better develop strategies to battle these antibiotic-resistant infections? So in the last section, we talked about how resistance often comes as a fitness cost. Thus, one strategy we could consider is, can we actually reverse the spread of resistance by either stopping or decreasing our use of antibiotics? So we talked about how in competition experiments in the lab, When we grow bacteria without any antibiotics, eventually resistant bacteria decrease in the population. So the idea is, could some similar effect happen when we stop using antibiotics in real life? But of course, this is happening in a test tube. It's kind of an artificial environment. So these fitness costs happen in the lab, but does this reversible resistance actually happen in reality? And the answer is not necessarily. So there have been some optimistic studies suggesting that this might work. So in Finland, in the 1990s, they had this big national campaign to reduce the antibiotic erythromycin. And this was a response to bacteria that were becoming resistant, and of particular concern was this bacteria called Streptococcus pyogenes. So you can see that starting around 1992, the red bar representing erythromycin really decreased dramatically. So the use of erythromycin really decreased dramatically over these 4 years. And then these researchers monitored resistance in specifically the S. pyogenes bacteria. So again, this is the time period that we're focusing on. And then when they looked at the percentage of S. pyogenes infections that were resistant to erythromycin, they actually saw that the resistant bacteria actually went down over these same 4 years. And this was kind of promising, and it was really touted as a success of this campaign to reduce antibiotic use. However, it's important to remember that correlation doesn't necessarily mean causation. So it's difficult to say whether this decrease in resistance was actually due to the decrease in antibiotic being used. And there's also been a number of other studies that have shown the complete opposite results. So for example, there's a study in the UK that was conducted around the same time period that showed that a really dramatic decrease in a class of antibiotics called sulfonamides did nothing to decrease resistance in the E. coli bacteria that they were monitoring. A recent study conducted in Sweden attempted to create a bit of a more controlled experiment to really define the impact of decreasing antibiotics. So they had an intervention county at the top and a control county at the bottom, and these graphs show the consumption of the use of antibiotics. So it's doses of antibiotic on the y-axis, and the time of the intervention took place over this, again, this 2-year period. You can see in the intervention country there's a really dramatic decline in the use of specifically this trimethoprim as the antibiotic that they were using. And in the control county, trimethoprim use didn't change. And then when they looked at the percentage of bacteria resistant to trimethoprim in infections, they saw in the intervention county there's a really modest decrease in resistance relative to the control country, but they concluded that this difference was really marginal and that ultimately the impact of decreasing antibiotic use didn't really have that much of an effect. So this seems to work maybe in some cases and not others. And why doesn't this always work exactly? We know that that resistance can often lead to a fitness cost, so why don't the sensitive bacteria eventually take over populations of natural bacteria? And one of the reasons is that, while resistance initially does lead to this fitness cost in a lot of cases, over time the bacteria can gain additional mutations that are able to offset these costs. And these are known as compensatory mutations because they're compensating for the costs of resistance. So let's return to that example we talked about earlier, where we saw that bacteria with mutations in the RNA polymerase that are resistant to rifampin have decreased RNA production. The same study showed that additional mutations in other parts of the RNA polymerase can actually restore the function of the protein. So the rest of this graph shows 2 bars that represent resistant bacteria that have 2 different compensatory mutations. So this bacteria has the resistance mutation, and it also has a compensatory mutation that partially restores its ability to produce RNA. And these bacteria actually have a different compensatory mutation that brings RNA production almost back to the normal levels of the sensitive bacteria. And the same study also showed that rifampin-resistant bacteria with these compensatory mutations also had restored growth. So if you remember, we saw earlier that the rifampin-resistant bacteria have these smaller colonies, meaning they're not growing as well. And then you can see that the bacteria with the compensatory mutations have larger colonies, indicating that their growth is being restored. And the same study also showed that in those competition assays, the fitness of these bacteria was also improved. So now we have bacteria that are still resistant to rifampin, but they also don't have that much of a fitness cost. They're almost as fit, and they can almost grow as well as the sensitive bacteria. So this is a problem if we want to try to reverse resistance. One lesson that we can take away from this is if we want to implement strategies to reverse resistance, it means that we have to act quickly before bacteria have a chance to get these compensatory mutations. So it's difficult to reverse resistance once it's already been established, but can we prevent bacteria from becoming resistant in the first place? Combination therapy is one really important strategy that we can actually use to prevent the development of resistance. And the idea behind this is it's less likely for a bacteria to develop resistance to 2 or more drugs as opposed to just one. If you can think about the analogy of a game of dice, where in order to win the game, you just have to get one number, such as a 3 on one die. You have a 1 in 6 chance. That's not too bad, but you have to get a combination of numbers on 2 die. Your chances are a bit slimmer. So this strategy of combination therapies played a really important role in slowing the spread of resistance. And it's used for a lot of important diseases. So, for example, tuberculosis treatment typically consists of 4 drugs for a period of 6 to 9 months. You'll see that one of these drugs is actually rifampin. Now, we know that it's fairly easy for bacteria to become resistant to rifampin, but when it's used in combination with these other drugs, resistance to rifampin becomes less likely. However, while this strategy can slow the development of resistance, it can't ultimately prevent it. So this graph shows the percentage of tuberculosis cases worldwide that are multi-drug resistant, so resistant to at least 2 of those 4 drugs that are usually used to treat it. So all the countries in blue, and the darker shades of blue, sometimes over 10 to even over 18% of tuberculosis infections are now multi-drug resistant. So combination therapy is an important way to delay the emergence of resistance, but eventually bacteria are going to be able to overcome this strategy. However, there are many labs researching whether it can make the combinations of drugs that we choose more effective. So for example, sometimes resistance to one drug can actually lead to increased sensitivity to another drug. To think about what this might look like, we've already talked about the situation where there's an antibiotic that can bind to a particular protein, but if that protein gets a mutation, the antibiotic can no longer bind. But what if there was a second antibiotic that wasn't able to bind to the original protein, but can actually bind really specifically to the resistant protein, and now this protein can actually inhibit or block the resistant protein. This is a really interesting strategy, and we call these compounds that can select against resistance because they specifically target the resistant bacteria, which have these resistance mutations in the proteins. And this is actually an area of research that I'm really interested in because I'm working with these types of strategies and thinking about resistance to drugs that we use to treat malaria. So I think it's pretty cool. And our lab and others have shown that using these kind of smarter combinations of compounds that specifically target resistant bacteria can actually decrease the rate at which resistance emerges. Now, these types of combinations haven't yet been tried out clinically, but there have been a lot of laboratory studies around this, and our lab and others have shown that eventually, even against these smarter combinations, eventually resistance will still arise. However, the hope is that using strategies such as this can at the very least buy some time. Another way that we might be able to specifically target resistant bacteria is by targeting some of those resistance mechanisms that we talked about, such as the Lactams enzymes or the tetrasulfone. And so if you remember, the beta-lactams enzyme, it actually is able to break down specific types of antibiotics. And beta-lactams drugs bind to these enzymes and prevent them from breaking down the antibiotics. And these types of drugs are actually currently used clinically to treat infections. They don't actually kill the bacteria, however, they make it so that the beta-lactam antibiotics that we'd like to be able to use are now still effective. So it makes it so that we can now treat bacteria with drugs such as penicillin, which they used to be resistant to. But the catch, and there seems to always be a catch, is that resistance to these drugs has also arisen. So when there's mutations in beta-lactamase, we can't target it anymore, and now we can't continue to use the penicillin or beta-lactamases in these situations. Now, the picture I'm painting here might seem a bit grim. It kind of seems that wherever we throw at them, bacteria are eventually able to find a way around it. But I want to end on a bit of a brighter note. So one strategy that we can always use against bacteria is to take measures to prevent infections. So this can be at a level of the hospital, so using infection control measures such as the proper protective equipment, masks, and gloves, and potentially isolating infected patients so that we don't allow resistant infections to spread from one patient to another. And there are also things that you and I can do at an individual level to prevent infections. This includes basic food hygiene measures like hand washing, as well as getting vaccinations. So there are several vaccines that already exist for bacterial diseases, including vaccines for tetanus, whooping cough, diphtheria. And then researchers are also working on creating new vaccines for other bacterial diseases. And prevention measures such as these are going to work whether a bacteria is resistant to an antibiotic or not. So they're a sure bet. And the other great thing is that if there are resistant bacteria, prevention measures can help slow the spread of those bacteria. So another optimistic note is that scientists are constantly researching brand new approaches to try to stop resistant bacteria. So some examples of these approaches include drugs that do not directly act on the bacteria themselves, but instead act in different ways to boost our own immune systems and fight the bacteria. Another kind of cool technology that's been developed out of the lab in Australia are these star-shaped nanoparticles. And these nanoparticles are able to integrate their way into the bacterial membrane and disrupt the membrane in a way that's less specific and that's less able to lead to resistance. So those are just a couple of examples. There's a lot of other cool ones out there. And I want to highlight that these types of studies are really important because, as shown by this chart from the CDC, the number of antibiotics that have been developed clinically and approved by the FDA has decreased dramatically since the 1980s. And so this really highlights the need for more academic research where, on the other hand, pharmaceutical companies may not have the incentives to invest in diseases that we already have medications to treat. But of course, the problem is those medications may not work a few decades down the road. So continuing research on new strategies for antibiotic resistance is really important. And I also think that just more basic research and understanding how resistance evolves is also important and can help us develop better therapies down the road. So to summarize, there are many potential strategies to combat resistance, but none are likely to be foolproof. We talked about how we may be able to take advantage of the fitness costs of resistance, although this can be difficult due to those compensatory mutations. We've also discussed how prevention measures are a very important way to slow the spread of resistant infections. And hopefully I've convinced you that biomedical research will be key to ensuring that we have new treatments for the future. As an overall summary, in part one, we learned that antibiotic resistance is a property of bacteria and that it occurs through this process of natural selection. In part 2, we discussed a variety of ways that bacteria can become resistant to antibiotics, and also how these resistant mechanisms can lead to a fitness cost. And finally, in part 3, we've talked about multiple potential strategies to address antibiotic resistance, but hopefully this also illustrates that it's a really difficult problem to tackle, and we'll probably need all of the strategies that I've talked about, as well as the new ones that we're able to come up with. Thank you so much, and I'll take any other questions.
So there's like certain environments where certain types of organisms are unable to live. Is there any way that we can make our body that environment place where bacteria is in?
It's a good question. My gut reaction is it would be difficult to do that and not kill us as well. But yeah, it's an interesting question.
Yeah, do you know if that lab in Australia are using the nanoparticles that found a way of making specific to bacteria, or do they just target all the cells in the area?
Good question. So, oh, I'm sorry. The question was whether the example of the nanoparticles, whether this wave was to be specific to bacterial cells. Actually, so they've done studies in mice where they show that these nanoparticles don't kill the mice, so they're not attacking the mouse cells. I think it has something to do with like the different size and structure of the bacterial cells compared to like mammalian cells. Of course, this is still a long way from being used in humans. It's still just in mice, and that's definitely an important point when considering any potential strategy to help type of drug or therapy. Yeah.
Is there a percentage on like how that bacteria is mutate?
Right, so the question is there a percentage of how likely the bacteria is to mutate? And so different species or different types of bacteria have different mutation rates. So it will depend on particular bacterial species. But yes, there is a sort of a particular rate, or average rate that each bacteria has.
The approach with vaccines is that is the approach to boost the immune system to be able to take care of bacteria?
Exactly. So vaccines work by. Oh, I'm sorry. I'm very bad at this. So the question was, vaccines work. Or the question was, what is the strategy for boosting the immune system? So I think I actually mentioned two. So there's vaccines, which the way that they act is by sort of either the vaccines give us either sort of a particular protein that's found on the bacterial, like the outside of the bacteria that our immune system can recognize, or some vaccines are actually like you put like either dead or weakened like bacteria or virus like directly into our bodies. And so those are different types of vaccines. And when we do this, in any case, it makes it so that we kind of prime our immune system to be able to now recognize those particular bacteria or viruses. And when we're able to specifically recognize them, we build up these proteins in our own body that can create a more sort of specific attack against that type of bacteria. And so that's why we have so many vaccines that we're given as a child, because each vaccine sort of allows our body to better recognize like a specific bacteria or a specific virus, and we get this really specific response against that disease. And then there's also drugs that people are working on that are maybe a little bit more general, so kind of boosting more general immunity within our bodies. But yeah, vaccines will provide us producing this very specific response and being able to recognize these bacteria.
What is the funding level that's being given? And you see this where until it's on people's doorsteps, they don't really call their legislators. And, it's places in the tip of Florida or in the Maldives where they're going underwater that they are the loudest voices for help on this issue. Will it take, I don't want to call it a tragedy or something, but like, will it take more harm happening for the public to fund this? Or is there enough interest within either companies or developing a drug that would be worth taking 4 to 5 years to have some kind of return on investment yet?
That's a great question.
Or like how are grants going,
Yeah, no, it's a good and sort of relevant question, and I'm not sure I'm fully equipped to answer that. Like my general sense is that there are a lot of labs doing this type of research, and like government organizations like the CDC or the NIH, which itself does a lot of research and also has a lot of research funding, like they definitely recognize that this is an issue, and I think there's more and more awareness. So I am, I think, like fairly optimistic that people are going to keep trying to, either do those studies with like the big bacteria plates, where we're really trying to get into understanding how resistance evolves, and or those other studies I mentioned at the end, trying to develop new technologies. Like I do think there is like a good amount of interest, certainly in sort of the academic level, and I think hopefully like it will also start to be adopted by pharmaceuticals as the need is continually recognized.
Yeah, I think that's very fair. I will maybe take this opportunity to say that drug development is likely the most lucrative sector of infectious disease research. So within infectious disease research, we're not necessarily stuff right there, but infectious disease as a whole is not doing super well in terms of funding compared to research for cancer, research for heart disease, the main killers of people in the developed world. That being said, drug resistance is not unique to infectious diseases. If any of you were here for the last lecture, we talked about how drug resistance can also be applicable to cancer, which is obviously a huge, huge problem. I think it also goes to say that infectious disease research is more than just drug development. We definitely need more funding for diseases, particularly in developing countries that get like, I'd say 1/100th the research, the funding that cancer gets, whereas this is a 100% true statistic. We could cure 1 billion people with infectious disease in the world if we gave them each 50 cents. But we are not doing that right now. That's my public health plug for the evening.
So it's the basic research to understand the diseases themselves, and not so much the need is for that plate and the time to let it grow and play around and just randomly find out correlation.
Yeah, absolutely.
I was assuming when I saw the plate that you do a little diagram with a little web of colonies in the plate, that whoever does that experiment can easily, they can sample each different part of the plate, and they have a way to take apart that particular bacteria and identify the particular thing that it gained.
Right, they sequence, yeah, you're talking about the video that we watched at the very end. The question was about the bacterial plate that we saw, and when they had the dots come up, you're exactly right, they sequenced each of those bacteria. So sequencing means that they kind of read whatever is like in their genome, so they're able to see the exact genetic changes that happen. And so
And they can tell whether that gave that particular population that thing that comes out or a missing core, or they can categorize it completely on your chart of mechanisms.
Yeah, that's a great question. So the question was, when they look at the genetic changes in that experiment, they categorize it based on the mechanism. And I would say I think the answer would be sometimes. So it depends. Like sometimes this is a type of bacteria that's been really widely studied, and we know a lot about its genes and their functions. And so, if there's a mutation in something that there's already been a study showing that creates resistance, or it's mutation in the gene that they kind of have a sense of what it does, then yes. However, in this and in other studies, oftentimes you might get a mutation in a gene and then a protein that we don't exactly know what it does, and then you need to kind of do further experiments to say, oh, we've seen that there's a mutation in this protein, and that seems to lead to resistance, but we don't know what this protein does. We should probably go and study that protein and find out more about it. And that's actually like the beginning of a lot of research studies. So that's actually an awesome question.
So they might generate new interesting mutations that they have to then go act out with knockout studies or whatever, to figure out what its function,
Exactly.
identify the function if they don't have a complete map already of all this.
Right. Exactly. That's actually a lot of what my research involves too in the malaria parasite. And that's that's something that we know a little bit less about. Its genes aren't as well characterized. So it's a problem and a challenge, but also something really interesting that can lead to a new project or a new interesting question. Happens. Questions? Okay, great. Thank you so much for your attention and for your questions.